In this video, we'll look at the three plant tissues. This first figure I show you has cross-sections of each of the three organ types in plants. So this is the cross-section of a leaf. This is the cross-section of a stem. This is the cross-section of a root. And you see that if we concentrate on the vascular tissue, where the vascular tissue is located pretty much dictates the pattern of tissue arrangement in each of the organs. You can see that in stems there is ground tissue both inside of and outside of this vascular tissue. In leaves you could see very discrete bundles of vascular tissue, but note here too in the stem that you've got discrete bundles of vascular tissue, maybe embedded within ground tissue and then eventually surrounded by dermal tissue. This is a little different than animal biology. Notice that the dermal tissue is only on the outside. You have dermal tissue lining your organs. So you have an intestinal dermal layer, and your heart is covered with a dermal layer. But in a plant, the only dermal tissue is on the surface. So vascular tissue in red, organized in bundles, except for in the root where it takes a central location in the structure. If we look more closely at these organ cross-sections, we note that we can divide the vascular bundles into the xylem and phloem tissue. Phloem tissue is operating under positive pressure and is used to distribute material from essentially any two spaces within the plant. The xylem, on the other hand, is a unidirectional transport system, and it is used to move water from the roots through the plant into the shoot and into the leaves, after which it often is lost to evapotranspiration into the environment. So you see that the vascular transport system is divided in each case into the red and blue. In each case, the blue represents the phloem, the red represents the xylem. Notice that in the bundles of the leaf, the xylem is facing up and the phloem is facing down. The difference between that and the stem is that in the stem, the xylem is facing in and the phloem is facing out. So in one case, you've got phloem closer to the surface, xylem further away from the surface. In the other case, you have the xylem and the phloem just simply being closer to different surfaces. So this isn't so confusing when you're looking at a nice flattened leaf like this. However, if you were looking at a circular leaf, and they do exist, you would not have a pattern that looked like that. Instead, you would have a pattern. Okay, I'm going to draw a circle into which I'm going to put vascular bundles. Xylem. What am I doing? I'm filling the upward facing subset of each bundle. And now I'll draw the phloem in here. Do you see the difference between these two circular structures? In one case, I've got the phloem on the outside, the xylem on the inside. In the other case, I've got the phloem on the bottom, the xylem on the top. Why would this be? Why is there this difference? Well, it comes from how these tissues are generated. Remember that the meristem is making the entire set of organs that are ever created. The vascular tissue is in bundles, and the vascular tissue is maturing from the oldest part of the plant up towards the youngest part of the plant. And each one of these bundles, when a leaf forms on the side. Here's a leaf forming, and there has to be an axillary bud. It's going to move and mature in that direction. And so what had been on the inside is now going to be on the top of that bundle. What had been on the outside is now going to be in the bottom. So what you're looking at is a structure that moves like that. What had been on the inside is the top. What had been on the outside is the bottom. That's why a leaf cross-section has xylem on the top, phloem on the bottom, whereas a stem cross-section has phloem on the outside, xylem on the inside. You might see that this makes it a little easier to identify ambiguous structures you might be handed in a lab or something. That one way to identify the difference between a leaf or a leaflet and a stem is the persistence, the existence of an axillary bud. Another way is to do a cross-section and stain for phloem and xylem tissue and see how those vascular bundles are arranged.